working on carotenoids, which are pigments giving its color to tomatoes or carrots, for example, and they have interesting properties as antioxidants, so they are commonly used in food industry or pharmaceutics. My research is about finding new ways of producing these molecules in microorganisms. So metabolic engineers see the cells, the microorganism, as a factory, and the molecule, the final product, is the result of a series of actions made by the workers, the enzymes. In this case, the assembly line to produce the molecule is already known, but maybe the efficiency can be increased. Here, for example, you have a right-hander just next to a left-hander that could interfere with each other. But if you move this worker on the other side of the assembly line, then maybe the average production might increase. Well, I'm trying the same kind of work in the cell. You just have to imagine a lot more of workers and molecules, and that the intermediate product could be lost between two enzymes, two workers. So the idea is to try to fuse the enzymes together so that the product go directly from one worker to another. And so, but this is basically it. I'm just showing like maybe different side of fusion for the enzymes, or just two different combination of enzymes. So this kind of research leads to the production of molecules for food or pharmaceutical industries. One famous example is the one of artemisinin, which is a molecule used in the treatment against paludism. Today, it is possible to produce this molecule in a yeast using genes from the Artemisia annua plant, where the molecule has been isolated in the first place. So I remember the first time I came to work, and I was really impressed about how Biopolis looks like. This is a huge campus with buildings that are dedicated to a specific field of research, but a lot of them are connected together. And I think it reflects the organization and efficiency about the research here in Singapore. Yes, the project I'm working on is part of the CNRS International Research Programs in Bioeco. So this IRP brings together labs from France and from Singapore. So in the French part, there are the Toulouse Biotechnology Institute and Toulouse White Biotechnology. And in the Singaporean part, there are the National University of Singapore and CEPI, the Food and Biotechnology Research Institute from ASTAR. So this work participates fully to the collaboration between Singaporean and French researchers through this PhD funded by the government agency ASTAR in Singapore and the Toulouse Biotechnology Institute in France through the INRAE grant, an INRAE grant. So first we were doing alternate team in the lab, 1 week team A, 1 week team B, and then the lab closed entirely. So this is when I started to conduct a review of the literature for a specific aspect of my subject. So this isn't something I was doing when I was in the rush of the experiment, but this is something that's going to be very helpful for the next phases of my project, because I will be more aware of what could work and what couldn't work. So I think this circuit breaker period is also an opportunity to learn and think about the way we work.